All right, I think we can at least get started now. So welcome again to DevoWormML, and thanks again to Vinay for last week's presentation. It was very enjoyable, and learned a lot about what he was doing. After his presentation, I thought, I wanted to make sure that people were I don't want to give like a super technical, approach to this. So here's Abraham. So I wanted to, highlight. Hi, Abraham. How are you? Okay. So I wanted to sort of highlight some things, and today I'm going to give a talk on something called input data. So, I wanted to kind of get at a little bit more basic level for people. So some people are, working in machine learning and some aren't. So I wanted to the experience. First of all, I'd like to ask, does anyone have any questions about, where we're going or any news or anything they'd like to share? Okay. We have a new member here, Aidan Rocke. He's worked in the OpenWorm Foundation in the past, and I've been talking with him for a while now on some various topics. So he's interested in machine learning. He's a mathematician by training, and so he probably have some things to add to this group. We were talking about a couple of machine learning topics in the recent past. So why don't you introduce yourself, Aidan, a little bit?
Right. So I studied mathematics at Aidan University, and that's where I met Balaji Getty, who did some behavioral neuroscience work for his PhD, and also was a director of the OpenWorm project. So that's how I know OpenWorm. And I did some consulting for machine learning companies the last 2 years, and recently I am very interested in developmental biology and its practical applications to medicine.
Okay. Yeah. Well, I hope you find it useful what we have here. And so anyone else have any comments they want to make? Anything they saw in the news? Maybe any milestones they'd like to share? Okay. So, all right. So why don't we get started? I have a lecture on input data that I'm going to present here. Let me share my screen. Okay. Can everyone see my screen?
Mm-hmm.
Okay. Present. So this topic is input data, and basically the idea here is that if you put data into a model or algorithm or whatever, it's going to reflect what comes out of it. And so I gave it a little Bayesian notation. It's cute. But it kind of means, there is a kind of conditional relationship there that if you put bad data in or something that isn't very good, you're not going to get a very good result. It does hold true, however, that if you put very good data in, you can still get a bad result. But if you want to talk about that some more in future weeks, we can do so. But our focus here today is this processing of the data, but to have a good, clean set of input data that we can then make inferences on. And so this figure is actually something from a cybernetics article I read, and I can't remember which article it was, but I think it's a nice graphic. So I wanted to introduce people, if you haven't visited this already, is the DevoZoo. And so I mentioned, I think in the first or second meeting, that DevoWorm has data that we've collected from different places. This is developmental data. It's largely embryogenesis data, although there are other examples in this repository. And so this is the link to the repository. And we've assembled a bunch of data sets that are, from different publications or from different sources. And they're labeled here. And you click through and you find it in a location and you download it. And you can use it for different things. You might use it for training a model. You might use it for analysis, whatever. And we've had a little bit of interest in it. I've used it for Google Summer of Code. I've used it for people who are interested in contributing to the OpenWorm Foundation. I've given them some data sets just kind of right here. So when we talk about input data, we're talking about various data sets that come from a place like this, where you can take like image data, or sometimes it's tabular data, and then put them into a model, and then find you can categorize it, you can build regression analyses, you can do other sorts of relationships with it. But there is a lot of open biological data. In fact, if you have been following the literature on this, you know that there's massive amounts of open data of very different types. And it's much more than people are actually using, because we have a lot of high throughput techniques, and we have a lot of people collecting data on different topics. And people, being scientists, they like to share with other scientists, so they make it available for people. And so if you want to get input data, one good way is to go to some of these repositories. And so we have a couple of examples here. EMBL, for example, is a good place to find molecular data. If you're interested in gene expression or sequence data, EMBL usually has some, and it's associated with a publication. So you can download it in tabular form, and it's, you have to do a little bit of research on what the variables mean sometimes, but you usually can get a nice data set that's maybe suits your needs. ImageJ has public data sets. So ImageJ is a image processing platform that is open source, and they are, it's run by the NIH. So they actually have public data sets that they use to calibrate their program for different algorithms that they use for image processing. Also, Kaggle, which is a machine learning company, Kaggle.com, they have a number of data sets that are open. So I gave this example here of avian vocalizations. This is some person who collected these data from, just sitting with a micro or a mic, and they've recorded birdsong in different places. And so, that data exists, and you can download that and analyze it. And vocalizations, it's a continuous type of data, so it's usually in some tabular form, but you are able to reconstruct waveforms from that. And then the SSBD database, which is run actually by someone who is collaborated with the DevoWorm group in the past, Ken Ho, who's at Rocke in Japan, and he runs SSBD database with some of his colleagues there. And this is a place where you can find a lot of developmental biology public data sets. So there are a lot of data sets for C. elegans, or for zebrafish, or for Drosophila, and they have data that's in tabular form, taken from cell tracking data, taken from other types of experimental setups. And so there are different types of open data, and I wanted to bring this up in case people were wondering, how do you get open data and use it for these models? But they all share this one problem, and that is that there's a criterion that we have to evaluate it on, or two criteria actually. One is the data quantifiable? So if we, for example, download an ImageJ data set, and it's a bunch of images, can we quantify those images so that we can use numbers instead of just throwing the image into the model? Also, if we throw the images into the model, can the model extract something that's quantitative or something that the computer can count. So that's something you have to think about, and that takes a bit of research on the researcher's part to figure out how they want to represent their data, or if it's something that they can easily deal with. And then, is it enough data? So in some of these instances we'll talk about today, is it enough data to do what you want, to make sure that the algorithm is trained properly and so forth. So I wanted to also bring this up this is from data science, and they have this term in data science called the 4 V's. And I sort of modified this for biology a bit. So there are 4 terms that all start with the letter V that are important attributes of data. And so the first of these is volume, and that is how much data is available. So, when you download one of these data sets, how much data is there for you to use, for you to train a model or whatever. This, maybe you're interested in a lot of samples, maybe you're just interested in quantity in terms of bytes. So, there's a lot of very high throughput data, so there are a lot of bytes. But in terms of useful samples, it might be a little tough to get, useful samples out of that. And so those are trade-offs that you have to think about. Another V word is velocity. And so this is something that, you say, what does velocity have to do with data? And it has to do with how much change over time does the data set capture. So if you're interested in movement of a cheetah, and you have images of a cheetah moving, but maybe you only have 6 images that are sort of sequential, of a cheetah running across the plain, can that really capture the cheetah's movement, or do you need much more data for that? Similarly, if you're looking at gene expression, and you want to know about fluctuations in gene expression over time, and you have 3 time points, is that enough data to really capture what you want? And so you have to keep that in mind when you get public data sets and work with them. Are you sampling it at the right number of samples per unit time? Secondary thing about velocity is that sometimes you think about velocity in terms of that question, but there's also this issue of velocity in terms of how fast does the field move. And so you might get a data set from 15 years ago, but it's maybe not relevant to some of the modern techniques. So maybe there's a better data set you could use. And then 2 more V words, variety, which is how much natural variation is captured by your data. And we'll talk a little bit about how you might be able to improve upon that later on. And then veracity, which is how reliable is the data before and after transformation. Veracity, of course, means truth. So can you ground truth or biology truth the data? If you have some data and it's just something that you've collected from a repository, does it actually match the measurements or other such data sets that are out there? And so that's a lot of V words to remember, but I think it's a good framework for sort of approaching trying to take data and then apply it to some machine learning model. So, how do we know if our data set is usable? And this paper that I have here in this slide, it lays out sort of a taxonomy. So, there's usability, how easy is it to figure out what's in the data set, the context of the data, the availability, the reliability, so is the data reliable, and the presentation quality. Those are all factors. And, this is, again, something you really want to evaluate your data set, your input data set very well before you use it. But, we're also interested in putting it into a machine learning model. And so in that sense, we want to make sure that the data is very usable and we understand what's in the data set, and we understand maybe how to label it or how to, maybe pool samples or whatever. But ultimately, we're looking at using this for training data. So we want to have something called training data to put into the machine learning model. which is a mathematical model or some algorithm. And then eventually we want to make a prediction. So we want to take, I think we saw this in the digital basilaria stuff, where we had some model here, and we took training data, which were microscopy images, and we applied it to this model, and then we made predictions about the shape of cells and their locations. So the training data is, of course, very important here, because if I want to train the model to make good predictions, the training data has to have, easily identifiable features, or in the case of the digital basilaria data, cells, individual cells that the machine can parse. And so this training data, then, all the stuff that I've mentioned just previous to this, is going to be very important for training a proper model that can make proper predictions. People think of machine learning sometimes as like magic, but it's really not magic. It's just you're taking data, you're taking a good model, and again, what model you choose is something that's, again, you have to do a lot of research on, but you just have to match these up, and then you can get a good prediction.
Bradley, could you please hide that little button at the bottom?
Okay, I'll hide it. All right, so yeah. Oops. So this is the link here to this article that I got this image from. So in case you want to look that up, and I'll make the slides available after the talk. And so remember this sort of framework, because this is the way, when I start getting into pre-trained models and things like that, it'll make a little bit more sense. So when I talk about training data, we could use open data, or sometimes for different tasks, they have specially, benchmarked and, assembled training data sets. So this is the MNIST database, and this is a famous benchmark data set for generic machine learning applications. So you can see it's a series of digits from 1 to 9, or from 0 to 9. And it's just people writing these numbers by hand. But notice all the variation here. Look at the row 4s, for example. You have in many different ways. People close this fork here, they keep it open, they write it in sort of a slanted manner. Here it looks like a Greek letter. This almost looks like a trident, but it's still a 4. And so forth. And so there's a lot of, the point of showing this is that the input data should have a lot of variation in it. Each of these columns is a sample, which is, something that's like an individual case. Okay, this is one case, and then this is another case. In a biological context, you might think of it as like individual organisms or something like that. And so you're just showing the variation in this training set. And you're lining it up, and you're giving the model all this data, just over and over, but different variations on it, so that the algorithm can pick out, say that this is a 4, but this is also a 4, and this is also a 4. And it would be hard if you just gave the machine this column for the machine then to say, oh yeah, this is a 4, even though it's trained on this 4. And so the same thing with 7s. Sometimes people put a cross through their 7s, and sometimes they don't. And so, again, the machine wouldn't know necessarily that 7s exist in both ways unless you show the machine that through this training set. And so this is a benchmarked set, meaning that it's something that people can use in publications and say, we trained it on MNIST database. And people know what that means, and it's always the same. And it always should give a very similar result. And so I think the thing to remember about this is that biology, in particular developmental biology, does not really have these types of data sets. We have some that are famous, but there isn't like a benchmark data set that people know, how it's going to perform when they put it into a machine learning model. But there are also other types of benchmark data sets for training. So if you're not interested in handwritten letters and numbers, we have celebrity faces. So there's a database where they just give it a bunch of faces that are of celebrities, and they train it on this. And you can, this in this case, you have a label, which is the name of a celebrity and a face, and then the algorithm, knowing all of that, can distinguish faces or can, identify faces. There's the Stanford Cars dataset, which is, again, different models of cars. And again, it's all about like the specific shape and features of the car, that if you present enough a variation to the model can distinguish between different models of cars. It's pretty simple. And then the same holds true for the Iris dataset. And the Iris dataset is interesting because it's actually a biological dataset that was created by R.A. Fisher like 100 years ago. And it's basically taking pictures of irises, or the flower iris, in many different versions of the flower. And so, you assemble this huge data set of, irises, different shapes, different variants, and you can identify different individual, or, even if you have something that looks odd, that doesn't really even look like an iris so much, but it has distinguishing features that allow the algorithm to pick up these features and correctly identify it as an iris.
Bradley, next question. In the case of the iris data set, how important is it to have a complementary set of flower pictures which are not irises?
Well, that's something I'm not going to talk about in this lecture, but yeah, you bring it up, I will mention that. So the iris data set is good for identifying irises, but when you start, and you have like a lot of variation there, but then when you start getting away from like the typical iris, the average iris, then the performance starts to degrade. So what people have done in the more recent past is they've come up with something called adversarial training. And so this is the idea that you train it on irises, but you also train the machine on things that aren't irises that might look similar, or they might be like, related things that aren't irises, because, you pointed out that, you can have variation in the data set, but if you present it with something that looks maybe something like it, but it's like a borderline case where the machine, it's really ambiguous to the machine whether or not it's an iris. Then you have this adversarial training set that you can use, say like, tulips, that basically, or, different varieties of flower where you have, different shapes that maybe are similar to the iris but aren't. So you can put a label on those things. And then eventually, the machine might lock on to something that maybe looks like an iris but isn't, but is. So it's like they can deal with those boundary cases a lot better. So yeah, the people have used adversarial training for that. So yeah, it is important to kind of give it other instances. And again, what you're going to end up looking at is how the sort of the success rate and the error rate of the model. So the success rate would be how often does it correctly identify an iris, and then the error rate is how often did it identify something that's not an iris as an iris. And so that's a topic that is very intricate, but I'm just giving you a high level view.
In the medical field, those would be called false positives and false negatives.
Yep. Yeah. I think they call them that in machine learning too. I just wanted to give a more colloquial example for people. So that's the idea. We want to be able to identify things that are cars or irises or celebrity faces, and we want to reject things that are not. And so, the same holds for like developmental biology. If we want to identify, like, say, a cell boundary, we want to find the cell boundary. We don't necessarily want to lock on to things that look like a boundary. we want to be able to like refine our search for features that are likely to be a cell boundary. And so now we get into something called pre-trained models. And so we have training data, which is input data, but we also have pre-trained models that we can use. And I mentioned this during the talk on digital basilaria. That's what we were using as a pre-trained model. And so the definition of this is an architecture that is validated by a benchmark data set, so the thing I just showed you, to solve a similar problem. And so pre-trained models are common in deep learning and nonlinear programming, which is a method for, usually it's used for linguistics research or text identification. But, that's not something we're going to talk about today. But so pre-trained models are reliant on that benchmark data set. And the architecture is like a neural network architecture that is like pre-weighted. So you find the weights and then you apply it to something that's similar. The question, of course, is what is a similar problem? And so a similar problem might be, if you train it on identifying cars, it might also work for, irises because you're looking at things with features, or it might be like you could create a pre-trained model just for some domain, like handwriting, and then it may be very good in that domain. But then the question is, what does similar mean? Can you transfer it to other problems? And that's sort of the downside of pre-trained models, is you don't know that. And so in some cases, the model may be generalizable to a host of problems. So there are some pre-trained models such as DeepNet or Mask RCNN, which are just two pre-trained models that exist in the deep learning literature. Those models are pretty well generalizable. People have used them on a wide range of problems and gotten pretty good results. But it's worth noting that if you have a pre-trained model, it may not be good for your problem domain. So, especially like in biology where we have a lot of variation and we have a lot of things that are moving or, unclear boundaries or whatever, that's going to be an issue to think about very hard before you think a pre-trained model is going to solve all your problems. And, in deep learning, again, they have this wide range of pre-trained models: ImageNet, ResNet, Inception, DGG. I mean, just there's a huge list, and the acronyms aren't important, but what's important is to recognize that a lot of these models exist, but not all of them are made for specific areas of interest or fields of research. And so, again, here I got two examples of publications that mention pre-trained models. One is an archive preprint, an analysis of deep neural network models for practical applications. So they talk a lot about pre-trained models in that. And then opportunities and obstacles for deep learning in biology and medicine. And then that actually is an example of pre-trained models for biology and medicine, but they don't really propose any specific pre-trained models. They just kind of go over the idea. So what I'm saying here is that there's a lot of opportunity to perhaps come up with pre-trained models for specific types of data. Pre-trained models also allow for clearly defined features and classes to be built from data of a specific type. So, you could have scenes, faces, cars, or even tomatoes. And, we have a lot of variation, but each of those types of things are very different in the way the variation is distributed. And so here's an article, it's a blog post on highlighting 10 pre-trained model types and the data sets that come with them. So, this is mostly non-biological stuff. And then overall, pre-trained models allow for faster training. So we use a pre-trained model so that we don't have to spend a lot of time training the model on input data. We can use some input data and then a pre-trained model, and it helps us get to where we want to be. But that's not always the case. So you might want to approach these models with caution. And here's Medium article discussing why that is. And again, why that is because you don't know, whereas a pre-trained model might be very good for identifying cars, it might not be good for identifying cells. And so that's, but pre-trained models are an option. So, and then the next thing I want to talk about beyond pre-trained models is data augmentation. So we have our input data, and we have, of course, we can't capture every instance of variation. And we know that like the machine doesn't know like how to automatically make mental transformations. It's not like the human brain in that way. It just knows what it sees. And so one of the things that people do is they augment their data set so that they can give the model a wider range of examples of what these things may look like in the real world. So if I were training my model on dogs, for example, pictures of dogs, the algorithm might pick a dog like this, sitting down, sitting at the right angle, facing forward. That's a dog. But then if I show the model a picture of a dog, kind of like sitting on a couch or at a weird angle because I took the picture at a weird angle, then it might not be able to identify the dog so much because it's relying on some of these cues, some of these vertical cues here that are lined up in a certain way. So what you can do to avoid that is create these augmented images. And so again, in this case with the dogs, you're just transforming the image in different orientations. And so now the model knows that dogs exist sort of at this angle, like straight up, but also if there are pictures where the dog is at an angle for whatever reason, or there's some skew in the way the dog sits, then it can identify the dog properly. And so, I mean, dogs are kind of a toy example, but you can imagine like other things, like even cars. If you took a picture of a car, the picture was at an angle, or the car was going up a hill, you might not be able to identify something as a car. But if you give it examples where it's rotated in different ways, it can pick up the variation of how those attributes it's using might line up. Another example is this picture of a giraffe, where we have just pictures of a giraffe that are taken in nature. They're photographs. But the thing is not just important to have the information about the giraffe, it's also important to have information about the shape and maybe the shape of the background. So what we can do is create masks of each base image, and we can train the algorithm on the masks as well. So this way now the algorithm has shape information, both of the object and the background. So it can identify the background elements and the foreground elements and then make a correct prediction. So this is just the citations of these images. But using data augmentation in this way, it allows you to kind of make up for some of the variation you don't have in the input data set. So if there's, not very much variation in your input data set, you might consider data augmentation to remedy that. And I think Vinay actually used some data augmentation strategies in his project this summer. And so it's important for biological work.
Yes, sir.
Yeah. So do you have anything to add on that, Vinay? No.
Yeah, basically, I also did that the same augmentation strategies like positional augmentation and color augmentation. The most basic thing that I concentrated is on brightness level, because in the raw, like when we consider real life data, when we take data from the cameras out there, mostly there's one factor that is we have to deal with, that is the brightness factor and the contrast factor. So I put in more focus on the brightness factor and contrast factor. In like I did more augmentation in that way. Like I took the input images, I increased the brightnesses up to like 10 levels each time and made different images, and I included them in the dataset, so that the deep learning model will understand even if the brightness level changes, it will understand that it should not consider brightness too much, because if I put one image and give a corresponding label, and if I increase the brightness level of that input image, and if I give the same label, then it will understand that brightness is not a big issue. So most of the real life data sets will be suffering from that fact only, brightness and contrast.
Yeah. Yeah, I think that's a good point. I think the point, though, of augmentation, too, is to really kind of give uh, your machine learning model a better sense of what the world, what the world looks like, but also to kind of help it along. If, like in some cases with microscopy data, you might not have very good information in that one level. You just know that that's what it looks like, but it might be hard for the algorithm to lock onto features, and this kind of pulls some of those features out um, at different brightnesses and things like that. So, and then this is a pretty complicated slide, but basically this is the idea of upsampling and downsampling. So now in your input data, you have samples of different classes or different types of data. So, in a biological data set, you might have animals of different, striping patterns, or you might have cells of different shapes that are, sort of in the same class of type of cell. And so these are sort of classes that you can label before you train the model. But also, kind of that the model will sort of lock onto these as natural categories. So, but your data aren't always evenly distributed in terms of these categories. So, you might have, like, if you might be looking at fibroblasts, which are a type of cell, and you have different shapes that they have. And then, of course, some shapes are going to be overrepresented in your input data set, and some are going to be underrepresented. And so what that means is that to give the machine a little bit of help in identifying each class, you can artificially enhance the number of samples in a given class. So upsampling means that you have a number of classes, and some of the classes you have very few examples of, and some classes you have a lot of examples of. And so you would upsample the ones that you have very few examples of just by giving them, doing things like data augmentation, or maybe just going and finding more samples from that class to balance out the data set, input data set, so that you have a similar number of samples from each class. And downsampling is the reverse of that. So maybe you have a lot of samples of a certain class because it's a very common cell type, but you don't really need that many samples to really train the model on what it looks like. So you might just get rid of a number of those samples and just even out your input data set so that it represents a number of samples evenly. So this is a schematic here where the original data set has this class. Maybe you have like 6 instances here, and in this class you have 60. Well, you want to do something so that you can make this equal in terms of its representation in the sample. So you might augment these data in different ways so that you can get to this even number of samples per class. And there's a practical reason for this, of course, and that is the machine won't identify some of these smaller classes if you just leave it unbalanced like that. So the algorithm is going to pick up on what it sees. And if it sees things in the class where you have a lot of examples, it may just identify the ones in this red category as the same thing because it doesn't know any better. It hasn't seen enough instances of this red class. And so you want to even out the number of samples in each class that you give the machine to train on. And so, again, this is another figure from Google. They explain it in terms of weighting each class by the number of samples. And so, you can approach this sort of systematically. You might calculate how many classes you have and how many samples of each class, and then figure out how much you need to upweight or downweight each one so that you have a uniform data set. And so again, these are the links to these images that I took, but also it contain some more information about these kind of strategies. So if you're interested in this topic, you can read these articles, and they should give you more information. Finally, I'd like to conclude with talking about synthetic and pseudo data. So I think we've talked about augmentation and balancing your data set. Now I'd like to talk about synthetic and pseudo data. And so this is something people use to, again, provide training data for their model that they may not otherwise be able to get. So the working definition of this is a model of data that generates something not found in the original measurement, or something that is not directly measurable. So we have examples such as interpolation between samples, dynamical modeling of a hypothetical regulatory process. So, like, suppose that we had two different types of images, and then we train the model on that. And of course, it can identify the two different types of images pretty well. But then we give it an input image that is in between the two, like maybe like a hybrid organism, then we'll be able to identify that sample is its own thing, or is it going to throw it into one category or another? So you would need to create some sort of synthetic data to give it information about that intermediate case. Again, even with like regulatory processes, you're not going to necessarily get all the data you need to look at something. So if you wanted to look at some molecular pathway where kind of the components, but you don't necessarily have all the data for each of those components, you might create a data set that sort of simulates that missing piece. And so there are at least three approaches to this. Some of this you may have encountered in statistics class, resampling of the data using methods such as jackknifing and bootstrapping, small sample inference, and then data-dependent priors, which is a Bayesian method specifically. Those are things you can look up on your own, but those are like basically the three approaches that are in the literature. So how you might make a pseudo data set, you create archetypical labels. So you would label these fake variables or fake instances. You might then use a distribution, like a Gaussian distribution or something, to create a series of plausible values. So you make an estimate and you say, my process is Gaussian, or it's normally distributed, it's random, so we'll generate these values, and this is what they might look like in the real world, and then that's how I'm going to model it. And then you might sample these fake data in ways that test hypotheses or allow for variation. So you might say, well, we know it generates these values, but there's some values that are more likely than others, so we can make some other assumptions. And from that, you have a nice synthetic data set that you can then use to train the model on. And then, of course, you can also use labeling in the same way. So they have something called pseudo labeling, which is used a lot in semi-supervised learning. So again, this is where you create labels from unlabeled data. You guess what the labels should be. You make them up, but it's an educated guess. And then you use the more formal labels that to take out false positives that maybe you didn't account for in your fake data that you've created. And so you can use synthetic data to sort of get a leg up on your model, but then you can also use the real information to filter out things that are obviously wrong. And so consider the following spherical systems. Here's a image or an animation of a C. elegans embryo in early embryogenesis. And this is a Drosophila embryo, and of course, they're very different in terms of how they're, the processes going on here. But if we just threw this into a machine learning algorithm, we may or may not be able to really describe this or make predictions. So this is a nice example because things are moving around. they don't have very clear boundaries a lot of times, and there are no labels, at least not in these images. And so, how do you deal with those using the strategies I just showed? It's just something to think about. I've also come up with some questions for application to developmental biology, and I think we've covered a lot of these, but just keep in mind that we don't have good training sets for developmental biology. So what would a good training set look like, and what properties should it have? And I just leave you with that thought, because I think that's probably the most important thing to remember about this, is that, on the one hand, we have very good methods for machine learning that are, emerging, but they're not really applied to developmental biology so much. So that's something that's why I kind of created this group, so we can think through these issues. So any questions?
Brian, in terms of the Gaussian distributions,
Yeah.
The tails are what people would call outliers.
Yeah.
Now, are you suggesting that one should weight the outliers so that they are detected?
Well, it depends on what you want to identify, I guess. If your data set, it has a lot of outliers in it, then you might want to weight those more.
A lot of them then.
Yeah.
Not outliers.
But I think if it's about in the data augmentation example, what people are using these for are, of course, distinct classes. So people are thinking more of they think of their data as having discrete states, and that's an assumption that may or may not be true. But you have these distinct things that you want to identify, and so the idea is you don't want to miss things that are maybe rare, but because the idea is that the machine doesn't know whether it's rare or not. It just knows if it sees it, and it needs to know how to classify it. So if it's rare and it comes up in the data set, the machine, of course, is going to just misidentify it, because it doesn't really know what the correct label should be. So you might want to, in a case like that, you might want to have more instances of outliers. You might want to give it a lot of instances of outliers so I can identify those. But the idea is that these things exist in discrete states, and that's kind of the problem too, is that you have a lot of things maybe in biology that are intermediate states, if things are changing, and so that's a problem.
Yeah, there's also an interesting problem here, and you introduced the idea of interpolation using a model, a regulatory model or something.
Yeah.
And basically what you're saying is we will create a model and then massage the data so it fits the model. How do you know if the model that you used is wrong?
Yeah, that's true. Yeah, it's that's why you have to do research before you apply this stuff. So
it's a booster, because you can't test the model without having the data.
Right.
So,
yeah.
Read the model into the data, and does that force the data to fit the model, or does it give you criteria for saying, I used the wrong model?
Yeah. Yeah, and this, of course, applies to mathematical modeling as well, right? So if you come up with a model and you say, well, this is the way we think it works, sometimes it's just not a very good model. So it's I guess it's hard to really, basically with regulatory stuff, people have been doing work with high throughput data, which is just gobs of data, and they've thrown it at a model and they've gotten outcomes. But sometimes you look at these papers and you're like, what are they do they think about the biology, or is it just throwing data at a model and seeing what comes out?
I guess what I'm trying to say is be a little more concrete. In developmental biology, there are many models of how development occurs, and some of these I regard as rather mythological. So the question is, if you take one of these models that might be completely wrong and use it to massage the data, somehow enhance the data. Are there criteria by which you can tell the model screwed things up?
Well, yeah, I mean, I don't know. There are no established methods. So I mean, no one's really published a paper saying this is how you would verify this model. I guess one way would just be to try different models, like maybe try something where, maybe that something is, making some sort of regulatory.
Okay.
Yeah. So like people.
Let me be very concrete.
Okay.
We're working with some fellows at Tennessee right now who wrote a paper on the French flag approach to as a model of developmental biology.
Yeah.
And they misquoted some of the work by Natalie and me. Oh. And the way we're resolving this is that we're writing a joint paper with them at the invitation of the editor of the journal on discussing the French flag model versus differentiation waves. Okay? Now, this is, of course, the paper is not going to be based on any kind of detailed machine language learning, but it's an example of trying to discuss two entirely different models for the same phenomenon.
Yeah.
Okay?
Yeah.
So that maybe I'll put you down as one of the referees.
Yeah. Yeah, that would be be interesting to see. Let's go to the comments and see what people made comments on during the. Okay, computer tomography, bad data, image out. Yeah, bad incomplete image. So yeah, again, your input data means everything here. Aidan and Dick were talking about the sync folders, and then please hide sharing button. Yeah. Okay, so that was. So anyone else have any other questions?
Let me elaborate on the computer tomography thing. It's in computer tomography, you're taking projection data and reconstructing an image. And what we found out in our early experience, if you put in very bad data, you still get an image out, and your brain tries to interpret the image rather than realize that it's nonsense. And so the old adage, garbage in, garbage out, does not apply.
Yeah.
It can be garbage in, image out, and then what do you got?
Yeah. Well, I guess I was assuming that the bad image was garbage. So, but that's true. You could get something that looks real, and
yeah, it looks good, even though it's nonsense.
Yeah.
But it's very much like the business of Rorschach tests or things where people see things that aren't there.
Yeah.
Okay? Because our brain is always trying to interpret an image and make sense. And if we run data through an algorithm and produce an image, we try to make sense of it rather than conclude that the data was lousy.
Yeah. Yeah. Aidan has a comment. If we're talking about predicting temporal events, we might need deep sequential models. So that's another technique that we didn't talk about in this talk, but so we need machine learning models that can also do causal inference. Exactly. So we need, so there are a lot of different options for doing things like that, especially with like, when you're talking about, we talked about just putting data into the model. So now we're talking about what you can do with models themselves, like, because there's a back end to it. So it's like you train the model and then it can do things down the line. It just doesn't classify things and put them into classes. You can do more sophisticated stuff.
Yeah, let me elaborate on that. The sequential need not be temporal. It could, for instance, be serial sections of the same embryo. And you can use having, let's say, segmented one layer in an embryo. You can use that possibly, in some cases, as a starting guess for the next layer.
Yeah.
Okay. So the z coordinate could be the temporal coordinate instead of the temporal coordinate could be important here.
Yeah.
To So you actually have a four-dimensional problem.
Yeah.
Okay.
Jesse and Abraham, I haven't heard from you. Did you like to add anything or have any comments? Okay. Okay, thanks. Yeah. And Jesse says thanks too. Yeah, so we're so our hour is almost up. We're at the top of the hour. Aidan, I talked with Aidan maybe about giving a talk in the next couple weeks on a topic that we were discussing. Can't remember what the name of it was. But we can schedule that in. Thanks, Abraham. Thanks for the feedback. Anyways, well, we can discuss that later and set that up. So again, if anyone wants to give a talk about something or even share something that they've seen in the research community, please bring it to the meeting. We would welcome, let me know in advance, and we'll put it on the agenda, and we can talk about it. So thanks for showing up, and I'm glad that everyone enjoyed the discussion. And if you have any questions over